Welcome, everybody. I'm going to talk slow for the first couple minutes while Matt lets people in from the waiting room. Thank you for coming. Many of us in the audience may recall 4 years ago, 2019, a softer, more naive age. Late October, Dr. Jane Lucas was actually here giving us her job talk and convincingly presenting the importance of soil microbe and invertebrate interactions for tropical forest function. And I'm not going to talk about the research she's going to talk about today, but she then spent a year before coming to Cary, where she started in 2021. She spent a year at the University of Idaho as a prestigious USDA postdoctoral associate, really initiating some of the work that she's growing here at Cary, which she came in the middle of the pandemic and has done a really excellent job growing a new research program, both for Cary and for Dr. Lucas, really integrating some interesting community outreach that again was new for Cary, reaching out to local agricultural farmer communities and engaging them in soil science in ways that I think we're going to hear more about today. And also really jumping in with some pretty strong leadership skills, helping Cary move forward with our strategic planning, and also getting engaged in education at Cary and reaching out with high school teachers in addition to the farming community. So I think that Dr. Lucas has been really impressive in the last 2 years since she's joined our community. And I'm really excited to hear the 4-year update on how her research has grown in that time. I will also share a couple of things that I've learned more personally since Jane joined our community. One, she is a quite competitive ultimate Frisbee player and is very active in our local Hudson Valley ultimate Frisbee competitive sports arena, which I didn't know existed, and is quite impressive. And I also have learned so much about the different items of clothing you could plug in and heat up. Because if there's any part of you that's cold, Jane is the person to go to ask about what kind of plug-in clothing might be appropriate. So with that, I will hand it off and look forward to your talk.
Thank you. All right. I sadly won't be talking about plug-in clothing or ultimate Frisbee today. And also, I hope it's not a boon to my Minnesotan style that I like to use the aid of heaters to keep warm. But all right, instead, today I am going to be talking to you about the work I have been doing looking at soils, especially here in the Hudson Valley, as well as some along the Northeast coast and also out west, and kind of understanding what does soil health look like in this era of global change. All right, to get us started, I want to take us back in time, about 100 years back, to London 1928. It's a time I'm sure you all think about constantly. Actually, this is a really important year. There's a lot of interesting stuff happening in London at this time. Importantly, women over the age of 21 are finally granted the ability to vote. But we also see that there's a lot of economic unrest, unprecedented levels of unemployment, and this really big natural disaster is about to happen. The River Thames is about to flood in this incredibly historic way. It causes catastrophic damage throughout the city. And it's at this backdrop of an interesting era that something is about to happen that's going to change the course of history. It's something that has affected all of our lives. And that's the work of a young Scottish researcher working on influenza and some other bacterial pathogens at St. Mary's Hospital. So this young researcher is working hard, spending long hours in the lab, plating a ton of bacteria and trying to understand how does this bacteria grow and what might affect its growth and maybe limit its growth. Now, some of us might be able to relate to this, but he's starting to feel overwhelmed and decides, I need to get away for a little while. I'm going to go to the countryside and take a bit of a vacation for 2 weeks. He maybe wasn't the cleanest researcher. He left his lab bench a bit dirty. And while he was out, he had these bacterial plates that had mold spores land on them. And they created, similar to this picture, these zones of inhibition. So when this young researcher came back from his vacation, he noticed that there were these zones of inhibition. And so maybe you all already know this story by now, but of course, this is the story of Alexander Fleming and how he so-called discovered penicillin in just 1928, not too long ago, relatively. Now, is often the case with these types of stories, he isn't really the first person to discover or understand penicillin. In fact, as much as over 5,000 years ago, we have histories of ancient Egyptian physicians. They would take moldy bread and put it inside the wounds to avoid infection. Seems a little counterintuitive, but it's at that time that they probably understood that these different types of microbes are capable of fighting off other types of infections. So in reality, we like to think of what this happened as Alexander Fleming's kind of rediscovery or the modern discovery of antibiotics as we know them now. All right, so why do I like to open with this? What do we learn from this story? Well, first and foremost, it's kind of how we discovered penicillin in its modern form, and it's ushered in this huge revolution of antibiotic use. And I also like to emphasize that this isn't that old of a story. You know, we've only had antibiotics in the way that we think of them for less than 100 years. I also think that this story tells us the importance of taking vacation, because if Fleming hadn't gone off to the countryside, he might have seen that mold land on his plates, thought it was contamination, thrown them away, and never saw that chance discovery. And then finally, I really like this because it's a great reminder to sometimes slow down with our science and be observant for not the expected result. And I really give Dr. Fleming some credit because I think he understands this, and he was kind of a humble guy. He says that penicillin started as a chance observation. My only merit is that I did not neglect the observation. So I open that up to all of us to sometimes slow down and recognize that some of the biggest discoveries might be things that we aren't expecting. So with this kind of rediscovery of penicillin, it's ushered in a pharmaceutical revolution. We are now in this kind of constantly increasing period of antibiotic use, pharmaceutical use. And while we think that this is, this huge improvement, it's caused, it's allowed our populations to grow dramatically. It's a really important medical revolution. What we often don't spend a lot of time thinking about is what is the impact that this pharmaceutical revolution is having in our environments. Now, some of you all were probably here just a few weeks ago when my colleague Emma Rosie really emphasized how this pharmaceutical revolution should be considered the way we think about other types of global change. So here in a great study Emma did with some colleagues, they looked at kind of these classic global change factors, things like nitrogen fertilizer, phosphorus, as well as our increasing world population, that general increase in CO2 in the atmosphere. And so we can see this general trend over the past 75 years of these big kind of increases. And if we put that next to the use of pharmaceuticals, and in particular, the use of antibiotics, we can see that these types of trends are really mimicking other global change factors. And so today I'm really going to focus on how the inputs of antibiotics in particular are affecting our environment. And Emma did a great job kind of highlighting some of these effects, especially in aquatic systems. But today I'm going to be talking much more about their effects in our terrestrial systems. And in particular, their effects in pasture lands and croplands and other types of kind of native grasslands. And one thing I always like to emphasize is that, many of us might not realize how frequently antibiotics come into the environment and in the very myriad of different ways that they enter. First and foremost, over 150 different types of crops use antibiotics as a common management strategy. Fruit in particular is really reliant on antifungals because fungal pathogens can be extremely detrimental to crop yields. So very frequently, we have these extended periods in the beginning of the growing season where those plants are sprayed with an antifungal. Of course, the other big consumer of antibiotics and ways that they can enter into the environment is through the livestock industry. And so today, most of my talk will focus on livestock-derived inputs of antibiotics. And the reason I'm particularly interested in livestock inputs is because about 73% of all of the antibiotics that we produce today go into livestock. And this is pretty common. You can imagine it's important for treating as well as preventing disease. So it's a very common practice on that kind of medical side. And we also know that antibiotics are used throughout the world as a growth promoter. It's less so happening here in the US as well as in Europe, but it's actually expected to grow over the next 50 years. So this consumption is continuing to increase. And while the use of antibiotics can be a really critical and important part of our livestock industry, it's important to remember that these compounds don't just stay inside the body of those organisms. In fact, a lot of the times, these antibiotics are given to an animal, and then they go in one end and go out the other. So here, this is a graph of 5 common types of antibiotics. And what you can see in the red is how much of that original dose that can be excreted through kind of mainly manure pathways. And then in yellow, you can see the metabolized but still biologically active product. And you can see it's quite variable, but sometimes almost 100% of that dose can go in one end and come out the other. And so you can imagine that this kind of creates a continuous input process for antibiotics to be entering into our ecosystems. So today, I'm going to talk to you about 3 kind of major projects that I'm working on. The first, really diving into just the basic question of how do these livestock-derived antibiotics affect our soil ecosystems? The second is really diving into the interactions between antibiotics and global change factors, because I think this is where there's a lot of really interesting work. And I also think that as we experience increasing global change, those interactions are going to be really important to understand. And then the last part of my talk is going to be focused on antibiotic resistance in the environment. This is because I think when you study antibiotics, it's really hard to also avoid getting into that antibiotic resistance world. Alrighty. So let's jump into this first study. And this study is going to be looking at how the input of livestock-derived antibiotics come into our soils and the impacts that has on the soil microbiome, as well as some really important functional capacities of our soil with a special focus on carbon cycling. So to answer this kind of question of what is the influence of livestock-derived antibiotics, colleagues and I have been conducting a multi-year study in Virginia, where we went to this prairie that hadn't had a recent cattle pasture, cattle grazing, and we created plots that had one of 4 treatments. We have our true controls. Those have nothing added to them. And we have some added with a normal manure and untreated manure. And then we got manure from cows that had their common cephalosporin antibiotic course or a pyridomycin course. As you can imagine, it was a really glamorous day to have to chase cows around and capture their manure before it fell on the ground. So that's the joy of this type of field work. But what I really want to emphasize is these are pretty common courses, but we're giving the antibiotics to the cow and then collecting the manure. We're not directly putting the antibiotics into the environment. We went through and we created these clear PVC acrylic boxes. And what that allowed us to do is pulse in 13C labeled CO2. And that way we could kind of follow where recently fixed carbon moved throughout these different treatments. We ran this study for 2.5 years. And then throughout that time, we went in, we collected soil, we took lots of different metrics. And importantly, we also looked at the microbial communities, both the bacterial and fungal communities across this entire experiment. So the first result I want to jump into is the influence of these different treatments on our CO2 production, or the amount of carbon being lost to CO2. And so what we have here is time on our x-axis, and then we have the amount of CO2 coming off of our plots, the carbon in a CO2 form. And so pyridomycin here is in blue, cephalosporin is in yellow, and control is in black, and then that jagged brown line is our annual temperature. And so what you can see pretty clearly is that in the middle of the year, in the common kind of grazing season, we see that the plots that are getting manure from cephalosporin treated cows have higher rates of CO2 production, more carbon being lost to the atmosphere with that pyridomycin input. And so to also kind of complement this, we looked at how is the carbon then being reflected below ground. And what we see is this general trend where when our soils have that antibiotic-laden manure, they have less carbon recently fixed in their soil. So we're getting this kind of decline in carbon retention when we have this antibiotic-laden manure. So we went in then and said, okay, we see that there are some functional changes. This is shifting the carbon cycle in pyridomycin in particular to be expelling more through CO2, not holding it onto the soil. So what does the microbiome look like in these soils at that time? And what we see here is a pretty strong result of the microbiome between our different treatments differs. And the one thing, so each of these plots represents a whole microbial community. But what I want you to focus on are these centroids with their error bars. This represents the average microbiome. And what we see here is that the control manure is significantly different than both of our pyridomycin and cephalosporin environments. And so what this is emphasizing is that, maybe not a big shock, when you have antibiotics, it's changing what bacteria are present in the soil, not just in the microbiome. So that's changing the soil microbiome. And pretty consistently, we see that the shifts are not just everything being decreased in the presence of antibiotics. We see that lots of different taxa of bacteria are actually increasing. And one I want to just put a special focus on is the Acinetobacter bacteria. Maybe you haven't heard of this, but this one is particularly concerning because we know that Acinetobacters are an emerging health concern. They can cause a blood-borne infection that's very similar to pneumonia. And we see that they're increasing pretty dramatically in the presence of our cephalosporin antibiotic manure. All right, so from this study, it shows that when you are providing these antibiotic courses to livestock, it is capable of then changing the microbiome of the soil in their pasture lands. And we're starting to see these trends where it's changing that carbon cycle, shifting where that CO2 is being released and keeping less carbon below ground. But as I mentioned, we ran this study for 2.5 years, and we see kind of an interesting trend at the end of our experiment. We can see, initially this big increase in carbon loss to CO2 in pyridomycin, but by the end of our experiment, this isn't significantly different anymore. So I'd say that's actually kind of a good sign. It seems like maybe our soils are capable of acclimating to this presence, and they're starting to look more like that control manure environment, not a big difference. And so we're really curious, okay, if this CO2 output is similar across our different treatments, are those communities also converging? And so when we look at the microbiome at this very kind of final time point, what we found is that the microbiome is no longer different. They used to be different in that 1-year time period, but after 2.5 years, they completely overlap. The composition is really similar. And so this gives us some hope that these communities are capable of acclimating, kind of reconverging with the control environment. And maybe the presence of these compounds is only really influential in that first initial year. But being good scientists, we really wanted to dive into this a little bit further. And we really wanted to know, are these communities really the same? And how would these now converged communities respond if we brought in a secondary stressor? And so what we did is at that 2.5-year mark, we went back to our soils and we took them out of the field and moved them into the lab to do a heat stress incubation for 60 days. So our 15 and 20 degrees treatments are representative of a pretty average temperature for those soils. It's not expected to be a stressful environment. However, when you start to get to that 25 and 30 degrees Celsius, those are pretty high levels of temperature. They're not things that our microbiomes would be seeing for extended period of time. So we really wanted to see how do these different soils, respond to this, pushing them towards this high end of heat stress. And what we see is this kind of really fascinating result, which I'm going to walk you through because it's kind of hard to see initially. But if you look at our cooler colors, the blue and the yellow, you can see that no matter what the shape, which represents our treatments, they completely overlap. They are still staying that same microbiome composition. However, when you start to push our soils to that high end of heat stress, you can see that the types of bacteria that are surviving are different depending on whether or not you had that historic presence of antibiotics in the environment. And so what we're kind of calling this is this idea of a cryptic legacy effect of stress. When we went to those soils out in the field, it seemed like they were the same. Those compositions were the same. When we keep them at a normal temperature, they're still overlapping. But now, when they've seen this previous stress, when they get hit with a second stressor, it pushes them into a different microbiome composition. And we also have some evidence to show that that changes their function as well. So I'm really interested in this idea of kind of cryptic legacy effects of stress. And I want to just highlight that I'm not the only person who's thinking about this. In fact, a really interesting study just came out in 2000, or this year, I guess, 23, where they took soil and they either exposed that soil to heat stress or they kept it unstressed. So it has a historic legacy of stress or not. And then they grew plants in those kind of past stress or future stress or no stress or high stress. And what they found is that when plants are grown in a stressful environment with the stressful exposed soils, they grew a lot better. And so it's this idea that maybe a stress legacy could select for microbial taxa that are adapted to that stress, and that can then confer benefits to other organisms living in that environment. And so some people call this a microbial legacy, other people call it microbial memory. And I'm really interested in kind of diving into this more and understanding the mechanisms behind why this might be occurring. Okay, so this study has kind of emphasized that when we're treating our livestock with antibiotics, it's capable of coming into the environment, changing that microbiome, changing the function of these systems, changing our carbon cycle. And I'd argue that it also makes it a little bit less easy to predict how they're going to respond to future change because we're seeing that divergence depending on different historic stress. So this story, kind of we're putting antibiotics in through the cow, and then we're taking them out and doing an incubation. And so for this next study, what I wanted to do is see what happens when you hit soils with simultaneous stressors. And in particular, what happens when you put manure separated from the antibiotics, we can kind of tease out that influence, and put them in the backdrop of both heat and moisture stress. So to get at this idea of what's happening with simultaneous stressors, we did a field experiment out in Idaho where we have a control environment, we have an antibiotic environment, manure, and then antibiotic plus manure. And we ran this over the course of the grazing season in Idaho in a field that had never experienced any agriculture before. And we took lots of different metrics over this course of this study, but first I want to focus on, again, that CO2 response. How is our carbon being cycling across these different treatments? And so here I'm going to show you a graph that has the days since our first treatment on the x-axis, and then the amount of CO2 being produced, which in a lot of ways, this is a good indicator of microbial activity. More CO2 often means more active microbes. All right, so for our first month, what we see is that when manure comes into the system, it creates this big jump in microbial activity. That's not a big surprise. Manure has its own microbiome, so it's bringing in tons of microbes. You'd expect that activity to increase. But we don't see any difference between our control and antibiotic environments initially, which is an interesting result. And then our manure alone, we see by the end of our month, it has reconverged. It's behaving quite similar to the both control and antibiotic environment. But interestingly, when we have both the manure and antibiotic present, it's staying significantly higher. It's keeping that microbial activity higher compared to the individual stressors. And so these dotted lines represent when we do our treatments. So let's see what happens through the rest of the summer. And what we can see here is that, again, initially when we add our treatment, our manure causes this big spike in activity. But hopefully what stands out to you all is that over the course of the summer, this activity has really dropped. These differences have completely gone away. And in reality, these soils are pretty dormant. This is very low microbial activity in here. And so why? What's going on here? Well, if you know much about this area of Idaho, in the middle of the summer, it starts to go into this long period of drought where, in the beginning of the summer, we're getting a decent amount of precipitation, moderate temperatures, but then through the rest of the summer, we just get this absolutely no water coming into the system and this really big sustained higher temperatures. And so what this emphasizes is that while these different treatments can cause some effects, these other global change factors, these other really important regulators of temperature and moisture are swamping out any of those effects. And I'm honestly a little surprised to see this result because manure it's got all these nutrients, it's got a whole microbiome, but it doesn't really matter that we're putting this whole big manure pat into our soil. If there's no moisture, if it's really hot, nothing's going to be happening. And so this makes me kind of think about this idea of maybe we need to start thinking about global change factors hierarchically, and that maybe something like moisture in particular for soils is going to be this master regulator. And you might be able to put other stressors in there. Manure could come in, antibiotics come in, and maybe they're just not going to be that harmful if you're in that period of extreme drought or low moisture conditions. And so this is a concept that I'm interested in kind of diving into more, and I hope my colleagues have some thoughts about whether or not it's worth thinking about these different factors hierarchically. Now, we also are really interested in another metric of carbon in our soils, and that's something called carbon use efficiency. If you haven't heard of this before, basically carbon use efficiency is how much energy does it take you to build your biomass? We want it to be easy, right? So if you're a microbe, we want it to not take a lot of energy, not a lot of resources to grow and duplicate. And so when you have high carbon use efficiency, that means that you can take in nutrients, grow, and you're not having to work that hard to grow. Low carbon use efficiency means the converse of that. And the reason people are really interested in this carbon use efficiency concept is that it has been shown to be maybe one of the best predictors of soil organic carbon stocks and long-term carbon storage pathways in our soils. So this was a paper that, again, just came out this year, and we can see that as microbial carbon use efficiency increases, there's a general increase in the amount of carbon. So when we we're trying to keep our carbon use efficiency high so that we can hopefully keep soil organic carbon stable for a long time. So how are our different treatments affecting carbon use efficiency? Well, consistently, we see that there's a difference across our different treatments. The antibiotic increases the mass-specific respiration, which I recognize is kind of, it's the opposite. So more mass-specific respiration means lower carbon use efficiency. So when we see this heightened amount of mass-specific respiration, we know that these are less efficient soils, and consistently the presence of antibiotics can increase that mass-specific respiration and decrease the efficiency. Okay, so we see that the manure and antibiotics are decreasing carbon use efficiency together. We also know that we're changing our carbon cycles, but what's happening to the actual communities in our soils throughout this treatment? So we went in, we looked at both the bacteria and fungi. Today I'm going to focus on the bacterial response. And so after 4 days, we see that you are getting a different microbiome in your soil across all these different treatments. Our control environment and our antibiotic are similar to each other, but they are actually significantly different. Our manure microbiome is quite different from the control and antibiotics. And then the antibiotics plus manure is kind of representing that nice intermediate of the two. So again, just emphasizing that the bacteria that are capable of surviving these different types of treatments are different across the different treatments. So this is after 4 days, but then what happens at the end of the study, 120 days later? Well, what we see here is that by the end of our study, the community in our control environment, in the antibiotics alone, and the manure alone has reconverged. It seems like those soils have kind of, again, they've maybe acclimated or kind of rebounded back to what we would say control. But consistently, we see that the manure plus antibiotic maintains a distinct microbiome. Different microbes are surviving. And so what this is starting to highlight to me is maybe that as soils experience more stressors, their ability to recover and kind of come back to maybe what we call normal is diminished. And so they're staying in this kind of novel compositional state. Now, soils are more than just microbes. I do recognize that. So we went in at the end of this study and we also looked at how these different treatments might be affecting our soil food webs. And I'll just show you a small little results here. And what we see here is that consistently, when antibiotics come into the ecosystem, they decrease the abundance of microbivores, things like millipedes, isopods, some of those collembola, primary consumers. But interestingly, we also see that the presence of antibiotics generally tends to increase the amount of predators in the environment. And whether or not this is a long-term result, I'm not sure yet. But what I think maybe is happening here is that you kill a bunch of those primary consumers. There's lots of food available. The predators can come in and snap them up. But I'm not sure exactly why this result is happening. All right, so from this initial study, we are seeing that as you get these kind of interacting global change factors, it is capable of disrupting our nutrient cycles. They're also capable of shifting the below ground food webs. We do see that as those stressors increase, the kind of recovery or bouncing back to control environment is diminished. And maybe importantly, we have this really strong hierarchical effect that when it's hot, when it's super dry, it doesn't matter that these big treatments are coming in. Our soils are really kind of going into a level of dormancy. Now, for those of you who know me here at Cary, you know that I'm continuing to dive into these questions of what's happening to our soils when they experience multiple interacting global change factors. And so I have this project up here at the Cary Institute called Stressed Out Soils. And this is a currently 4-factor, hoping to be soon 5-factor study, where we are trying to look at all of these different interacting factors at the same time. So some of our plots are getting warming, some of them are getting a simulated drought, others are getting antibacterials, and others are getting antifungals, and then a bunch of the different combinations. So it's a fully factorial experiment. And before I jump into some of our first results from that, I do have to just say, this whole experiment has been such a team effort, and I really want to thank people here at Cary, in particular, the Cary crew. Mike Fargione has to be out there all the time when we treat them because he is our certified pesticide expert, as well as Fred's crew has been hugely monumental in getting these all these different structures built and maintained. So big thanks to them. And we've also had a lot of local students come by And that's been a really big help for us. And then, of course, individuals in my lab have really made a lot of this work happen. All right, so we are definitely still in the preliminary stages of getting this data. We're just finishing basically 1 year post. But I want to show you a little bit of how our different treatments are affecting the amount of CO2, so our carbon cycles. And there's going to be a ton of data on these slides, but what I am showing you here is the change in CO2 production over this past summer compared to our control plots. So if the bars are up here, it means they're producing more CO2 than our control environments. And if they're down here, it means they're producing less CO2. So what we see when we look at our individual factors is that fungicide, drought, and warming are all decreasing the amount of CO2 coming off of our plots. Some of this is unexpected to me. I kind of would have thought that the warming would have increased, but we'll dive into why that might be later on. And then we also see that consistently our bactericidal plots have more CO2 coming off. And so this is kind of that question of whether or not this is maybe that mass-specific respiration where the microbes that are capable of surviving have to produce more CO2 to persist in this environment. All right, so those are single factor plots. This is what happens when we have two factors together. We can see that maybe one of the most dramatic results is that our drought and fungicide plots have the biggest suppression of microbial activity of CO2 production. But interestingly, when you have warming and the bactericide, it's increasing that CO2 production pretty high above our control environments. And this maybe wouldn't have been predicted if you put together the individual single factors. They're not canceling each other out. They're kind of coming together to magnify an effect. And when we look at three factors, again, we're starting to see some surprising results where while warming, drought, and fungicide all alone cause decreases in CO2, when you put them all together, they actually increase the amount of CO2 being produced. And then finally, with four factors, we're just getting this general suppression. But again, our strongest suppression is happening in that drought and fungicide. So as I mentioned, these are just very preliminary results. There's a ton more to dive into. But I want to say that, we are seeing consistent changes in the CO2 across our different treatments. There's lots of fun questions to ask about why. We're also seeing some preliminary indicators of above ground vegetation differing across these different plots. One thing that is consistently showing up is we have the strongest differences among our different treatments in the buffer season. So really that kind of May time period, as well as the end of September to now. There's some maybe interesting reasons for that. And then I think the big take home point is that these treatments are not following a consistent additive pattern. And this kind of connects with some other work that individuals are doing that highlights that when you have more global change factors, our ability to predict the functional response is going to get harder. It's not going to be this consistent, like, this plus this is going to equal X. It's going to be this kind of interactive and multiplicative effects. And I'm really excited to kind of keep diving into why this is happening. Alrighty. So that's end of chapter 2. And so for the final bit here, I want to take a little bit of time, shift our focus a little bit by looking at how antibiotic resistance is occurring in the environment and how human activity and global change might be affecting the prevalence of this very kind of common but underappreciated environmental element. So many of us know that antibiotic resistance is a problem. The UN always highlights it's this global health emergency. And while we know that it's this really big emergency that potentially has the ability to affect anyone and anywhere, we really have a limited understanding of antibiotic resistance outside of a very medical individual person context. And I think it's important to recognize that antibiotic resistance is a very common thing. It happens in the environment, and it happens in all different types of environments. So of course, we know antibiotic resistance can occur in a hospital setting, but it can also happen in our houses. It can happen on airplanes. And of course, it's also part of our natural environment. So first and foremost, it's okay that antibiotic resistance exists. In fact, that's a good indicator of microbial function. So it's not necessarily a bad thing that antibiotic resistance exists in the environment. But what is consistently surprising to me is I don't think anyone anywhere could go to a field and pick up that soil and say, this is a normal profile of antibiotic resistance. This is what we expect to see here. It's this kind of black box. We know it's there, but we don't know how much or what types of resistance exist in the environment. And so this is something that I'm starting to kind of jump into and trying to understand, what is baseline environmental resistance? What does that look like? What's normal? What's average? And when are we starting to get into levels that we should be concerned about for both kind of like human animal health contexts? And then similarly, I'm really interested in how human activity might be affecting these baselines, and are they pushing those levels up into concerning levels, or maybe actually suppressing levels of antibiotic resistance in the environment. And so to explore these questions, we've been conducting this long, arduous study. I'm excited we're getting to the end of it, but we have gone to 220 soil sites around the United States. And as you can see, these have been classified into cultivated ecosystems or uncultivated. Cultivated really just means kind of a strong human presence. So this is croplands, pasture lands, but it also represents residential areas, so places like human lawns. And then the uncultivated areas are something like a native prairie or a forested system, areas that we think have less direct human input. And what we did is we used a technique that I won't get into, but it's a cool technique called targeted gene capture. It's a type of metagenomic technique that allows us to look at all of the antibiotic resistant genes in that environment. So we have about 80,000 different targets. And this really allows us to look at the whole profile of antibiotic resistance, as opposed to just choosing a few select genes to focus on. So the first question we have with this study is, how does antibiotic resistance vary biogeographically? And I acknowledge that this is going to be just a US-based study, so there's a lot more to jump into. But across the United States, we can see that the average richness, so the different types of antibiotic resistance in the environment, can be quite different. Here, the more red represents you have more types of antibiotic resistant genes. So that's like a beta-lactam and an aminoglycoside type of resistance. And so you can see that here in kind of the Midwest, as well as somewhat in the East, Northeast, we have higher average richness as opposed to some of these areas in the Southwest. We also see pretty strong differences in the total amount, the abundance of these genes in the environment. So here, those warmer colors represent higher abundance, and this is the total amount of antibiotic resistant gene copies. And you can again see that kind of our Midwest as well as Northeast has higher average amounts of antibiotic resistance in the environment, as opposed to some of our more Southwest shapes or states. But I think the big take-home point here is that it's really variable. It's not consistent across these different sites. And so a big question we have is, well, what's driving this variability? Why is antibiotic resistance really high in some areas and not so high in others? And then we're also really curious, can we use ecology to maybe make some inferences about why this resistance might be higher or lower somewhere? And so we have tons of metrics that I could spend all day talking to you all about, but I'm just going to show you one example of a result that I think is pretty interesting. And so what we see, or one of the things that we predict, is that antibiotic resistance is often considered to be an indicator of competition. Microbes will invest in antibiotic resistant genes because they're trying to defend themselves against competitors who are producing antibiotics. So if that's the case, then we think areas that have high competition should also have high antibiotic resistance. And we think that areas that have high microbial competition might be areas with more different types of microbes. So our general hypothesis is that as microbial richness increases, so too should the total amount of antibiotic resistance in the environment. And while it's not this perfect one-to-one signal, and we need to get into a little bit more of the analysis here, we are seeing that consistently higher microbial richness also has higher rates of antibiotic resistance. And so maybe that's an indicator that antibiotic resistance is associated with microbial competition. We also have tons of metrics about the biogeochemistry, and we see some difference, consistently. More metals in the environment has higher rates of antibiotic resistance. That's a pretty common result, but still lots of data to get through. So we see some of these general trends, but to get to that second question I had, I'm also really interested in how anthropogenic activity might be shifting some of these patterns. And so one of the big results that we see is that consistently our cultivated ecosystems, areas with high human presence, have about 47% more antibiotic resistance on average than our uncultivated systems. And I think it's really interesting when we start to break down what these different environments look like. And so what we see here is that there is variability across an ecozone, an ecotype, but consistently we see that areas like our drylands, shrublands, boreal forest soils all have lower levels of antibiotic resistance. What I think is happening here is these are kind of generally harsher abiotic conditions. So there's this kind of tolerance to competitive trade-off that we're starting to explore. But hopefully, and maybe concerningly, one result that stands out to you all is that the area that has the highest amount of antibiotic resistance is residential lawns. And this is not what I would have predicted. I would have thought pasture lands, because we know that's where we're using antibiotics pretty frequently with livestock. But consistently, it seems to be our backyards that have highest rates of antibiotic resistance. Don't know why yet. I would love to know why. I think I have some thoughts about it, but I think this is an interesting result and obviously maybe of important human health concern. Now, another thing that I really like about this technique that we're working with is that it can tell us not just how much is there, but also the different types. There's a lot of data on here. What all these different colors are is they're just different classes of antibiotic resistance. And what I want to show is something like our dry land. You can see the different colors. It means that the types of resistance that exist in dry lands are consistently different from something like our grassland or our pasture land. And so there's lots of data to kind of dive into here. But the reason why I think this is an important kind of resource is that if we can start to get an idea of what the average resistance profile is of an environment, then we can maybe develop tools for mitigating that potential disease risk to make that more targeted. For example, if lawns never have beta-lactam resistance, then maybe we don't need to focus on developing beta-lactam strategies in that area. There's a ton of kind of interesting human health connections to get into here, we're just starting to scratch the surface. Now, the very last part of this work that I'm kind of really excited about and interested in is bringing it back to these ecological questions and kind of some of the things we were looking at in my first studies. And one question we have is, what is the cost of having antibiotic resistance? We think that it can be a metabolically expensive tool to have. It takes extra energy to have resistance. We know that's probably the case because microbes will drop resistance if they don't need it. And that makes sense, right? Why hold on to something that requires more energy if you don't need it? And I've been working with a student at the University of Idaho, and we've been running these studies. He's treated different areas with different types of antibiotics. But the main result that I want to highlight here is that what we're seeing is that as antibiotic resistance increases, this is for the beta-lactam resistance, we also see that the mass-specific respiration in our soils increases. And so this is showing that maybe when soils have to hold on to antibiotic resistance, they're getting a trade-off and they become less carbon efficient. And so obviously that can have some long-term implications, that if all of a sudden our soils are constantly having to overcompensate for this resistance, they're going to be less likely to hold on to carbon for the long term. And I'm excited to keep kind of exploring these ideas, in particular, kind of what is the ecosystem cost of tolerance, and how does antimicrobial resistance influence our food webs. So there's a study that, or a project that I'm starting with individuals at the University of Connecticut, as well as SUNY ESF. And we're going to be looking at how waterways that are near agricultural fields, we see in our preliminary studies, these are different types of antibiotic residues. And what this is just here to show you is that throughout the year, at any point in time, antibiotic residues can be found in aquatic systems, and you can get lots of different types. And so what does it mean to be an organism that has to live in this environment that's constantly getting these antibiotics being pushed into the system? And does that kind of carbon use, metabolic efficiency trade-off scale up our food webs? We see it in the microbes, but does it also get transferred up into something like the mayfly and other higher trophic levels? So ultimately here, this study is just emphasizing that first and foremost, more diverse soils tend to have more antibiotic resistance. We also know that antibiotic resistance can vary quite a bit biogeographically, but that cultivated or human influenced systems have a pretty elevated amount, with lawns kind of being this extra hot spot. And then we're also starting to see these trends that soils that have antibiotic resistance are less carbon use efficient. And before I end, I do want to just say that there is some good news. It's not all doom and gloom when it comes to antibiotic resistance. And there's some studies that are starting to figure out, how do we maybe get resistance out of the soil? And what we see is that when you have earthworms in the soil, it always comes back to the earthworms. They are really good at digesting the soil and actually eliminating both the prevalence of antibiotics, the actual compound, and antibiotic resistance. So maybe earthworms are going to be, again, something we lean on to help clear out antibiotic resistance in areas that we think of as really highly contaminated. All right, so just a very brief summary. I just wanted to end, and this is a little bit of a shout out to my colleagues to just give you a sense of what's going on in my head most of the time, and hopefully things we can talk about more. But what I'm consistently thinking about is, first and foremost, these interacting global stressors and what that's going to mean as we try to develop management plans. Similarly, I'm interested in this idea of hierarchies of global change, and do we need to worry first about certain type of management for certain types of factors, and then other things can come into line. I'm also really interested in this kind of microbial memory and cryptic legacy effects of stress. And then finally, I'm excited about kind of exploring the world of carbon costs for being tolerant in a constantly polluted environment. And so hopefully I've convinced you all that as these compounds come into the environment, they have the potential to change the communities that live there as well as their functions. And I think that this has implications in that very One Health way, where they connect to humans, they connect to animal health, and they also connect to environmental health. And so hopefully that was exciting for you all, and I would be more than happy to take any questions that you have. Yes, Sarah. Thanks. Thanks. Sure, yeah. So Sarah asked a question about how these different interacting factors and are going to be affecting the soil, and then what that means for management decisions and carbon storage, and especially in the context of us developing maybe carbon credit systems. Yeah, it's actually something I spent a lot of time thinking about. I hate that the easy and frustrating answer is that I feel like we're running to solutions before we have the answers that maybe we need. But I also kind of go back and forth, which is like, it's okay to maybe be wrong for a while if we're making some progress towards carbon sequestration. So I spend a lot of time thinking about soils. We know that there's a lot of pushes to use soil as this, can we maximize soil carbon storage to solve our kind of global change. I think that the problem and difficulty and maybe the take-home point is that a lot of the management strategies are going to be very localized or site specific, and it's going to be hard for us to create these just general sweeping patterns of this is a good management strategy. So for something like the ag systems, we always talk about like no-till, and it's like, well, no-till is going to have more benefits on soil carbon storage in certain environments than others. And so I think what it emphasizes is that certain portions of the globe are going to have different types of global change occurring, and they're going to need different management strategies for rewarding carbon storage. And I think that's hard because we want these like big swooping answers to that. But I think it's going to actually just mean we need more regional work to understand what management strategies make the most sense. Josh.
So based on that question and answer, do you think it would be possible sometime in the relatively near future to actually rank these different impacts and say, no-till is nice, and it has lots of other benefits, but for carbon storage, the most important thing is A, the second most important thing is B, by summing up across all the agricultural land.
Yeah. So Josh asked if it's possible to rank management adaptation strategies as this is going to be most important, this is most, then subsequent. Yes, I think people are doing that. I mean, I think the fact that we talk about no-till a lot is an indicator that it generally tends to be this kind of holy grail, but it's complicated. I think we can to some extent. I think that if you are a farmer who wants to maximize carbon storage and you also want to maximize yield, certain areas might see more benefits than others, and that's probably gonna be really frustrating. But I think we can probably come up with these broad categories. It's just that it's gonna be a lot harder to come to the individual and then say, hey, your odds of getting a really great benefit from this carbon smart practice might not be as good as the person who's a couple states over. And so we have to be realistic about that stuff. Yeah, Shannon.
I'm going to jump in with some questions from online because everyone else here can continue to talk to Jane over lunch. So we have one interesting presentation. I'm curious if you have looked at the realistic load compared to your study loading levels. CFOs are not known for their limited impacts.
So I'm guessing that they mean the load of
So can you discuss the potential impacts of antibiotics on the scale that CFOs are using them and the waste loads they are producing?
Yes. So I think that I need to spend some time doing some calculations on that. All of the studies that we are running, I think are extreme underestimate because I'm going for introduction rates that are indicative of 1 cow, 1 manure pat. So when we were doing that first field study, we're taking the manure from the equivalent of just an average individual cow going out into pasture. So I think that when we have these areas that are very concentrated with high use of some of these compounds, their potential to cause like much higher levels of changes in carbon cycling, probably much larger changes in that microbiome. That being said, there's probably some limit, right? At some point, some of these systems are probably not going to, it's not going to maybe scale in a one-to-one way. We might eventually see some kind of leveling off of, decreases in carbon storage. So I think it, on the negative side, some of our results are definite underestimates, but on the positive side, maybe having those really concentrated areas affecting a small plot of land is better than spreading it across all of the United States or the world. So
one more question
online,
and then we're going to break, and people in the room are welcome to come for lunch and continue to ask Dr. Lucas questions whenever you can find her. But the last question, the last couple questions are about the idea of that composition of kind of anthropogenic caused genes versus your natural genes, and whether or not things like the lawn are because of high use, however you might determine that, by people.
Yeah, that is the big question. I think, you know, right now we're at correlational conclusions. So the next step is getting more into these causational studies, especially the lawn. I mean, we are seeing a lot of human input, a lot of human traffic, a lot of other organisms. They're pretty small. They're also highly managed, so they might be constantly being disturbed. You know, we're coming through with a lawnmower, we're coming in with fertilizer, so that kind of types of management. And it might not just be the physical presence of humans, but it might be the constant human activity that's turning over, maybe nutrient enriching the environment, that might be driving some of those patterns.
Thank you. Well, it is noon. Thank you so much for a great talk.
Yeah. Thanks, y'all.